Давайте, спасибо большое. Хорошо, спасибо. Уже со звуком все, да? Так, а ну-ка слайды. Слайды двигаются, все, могу, да? Хорошо, большущее вам спасибо, Дима. Это 7-я аудитория. О, уже тут студенты есть. Спасибо большое. Guys, do you hear me? Good morning. So, as usually, we have some delay because of some technical problems in the spreading of the lectures. So, but please, those who are there, because I use a different location, and please let me know whether you hear me very well or not. And should I speak louder or should I speak not so loud? Please let me know. I can see your comments on the screen, please. Tell me whether you hear me very well or not. Guys, could you tell me please whether yes, you can hear, right? Sangam. Oh, someone, please tell me that you hear me very well. Thank you very much, guys. I start now, and today's lecture is devoted to our current virus of our new concern, that is the SARS coronavirus type 2 and the disease it causes, COVID-19. And at the very first screen, or at the very first slide of our lecture, you can see one of the most current or new electron photograph of the coronavirus. And of course, you've seen some of those pictures in the internet, but today we studied step by step. And you can see here the virion of the SARS-CoV-2 with the diameter ranging from 60 to 140 nanometers. And you can see the very specific feature of the coronavirus. The outer space studded with the distinctive 9 to 12 nanometers long spikes. You can see them here, those spikes. And you know that the spikes give rise, the appearance of these spikes give rise to the name of the coronavirus, because the spikes give the appearance to the virus as a solar crown or solar corona, not king's crown, but solar crown. And you can see this is a sun and this is a crown around the sun, or corona around the sun. So that is the appearance of the virus. And before we discuss the new coronavirus, I would like to demonstrate a bit of the history of this virus's appearance, okay? And the history is that first those viruses, human coronaviruses, had been discovered in the mid-60s of the 20th century. So before we knew some animal coronaviruses, and they didn't study it that much. But then when the first human viruses had been identified, they had been given particular names. You can see names. I am not going to pronounce it. It's not important. But what is the important? The volunteers had been infected with these new coronaviruses because from the very beginning it had been considered that they don't cause very severe infections. So then, when volunteers had been infected, it results in the very mild disease, like common cold, maybe sometimes with some um, gastrointestinal disturbance. But then the disease was so weak and so mild that it had been considered then a relatively harmless respiratory pathogen. So, in fact, we forget about this pathogen. And I didn't see any important research works on it. So that was over for about 30, 40 years. And then take a look at this image, right? And this is the photograph from the newspaper from 2002, when really first dangerous coronavirus had been developed and spread within the human population. And the disease caused by this new coronavirus had been called severe acute respiratory symptom, SARS. And the virus that caused this had been called SARS-CoV. So it was introduced into the human population in the early 202, and it originated again in China. And this virus caused a very severe respiratory illness with some relatively high morbidity for respiratory viruses and relatively high mortality. So, uh, SARS had been considered as one of the latest in a series of new human infectious diseases. So, SARS is a zoonotic deadly form of pneumonia. So, and it emerged in southern China later in 2002 and rapidly spread to different areas of the Far East and several countries around the globe. And it had been spreading through the global air travels. They facilitated very much the transcontinental spreading of the new virus. And it was stopped also because of the measures that had been performed in the airports to stop spreading of the virus. But then, back in 2002, SARS caused very severe restrictions on international travel. Of course, they were not that like we have today's restriction. But then, that time, it caused the economic downturn in affected countries. And the World Health Organization officially declared the 16th of April of 2003 that SARS-associated coronavirus, never before seen in human, is the cause of the SARS. So it had been proved by WHO, I would like to repeat, in 2003. Then, what was the origin uh, of the SARS-CoV? Of course, as usually, it was a virus of animal origin. And the coronaviruses, with 99% sequence similarity to the surface spike protein of the human virus, had been isolated in China from a very cute animal. This is masked palm civet, Paguma larvata. And this is a cat-like animal that is used closely related to mongoose, and this wet cat is used as the delicious meal in China, so they used to eat it. And it is believed that humans become infected as they raised and slaughtered the animals and maybe eat undercooked. So that was the origin of SARS-CoV. Then the second virus appeared from coronavirus family was so-called MERS coronavirus. So that was second pandemic coronavirus. MERS, it means Middle East Respiratory Syndrome. In other words, it had been called camel flu or COVID-12 because 12 indicates the 2000 that it appeared in 2012. So this is the infection COVID-12 caused by Middle East Respiratory Syndrome related coronavirus. So there's a MERS-CoV. That was also a new coronavirus that caused infection in main Saudi Arabia. Initially, it had been called Saudi SARS before it was differentiated with SARS. And Saudi Arabia was most severely affected. Then also it caused infections in Jordan, Egypt, and South Korea. Since 2012, 27 countries have reported cases of MERS. And until the 23rd of May 2013, MERS COVID had not been differentiated fully with the SARS-CoV. And it had been called, as I said, we refer to it as Saudi SARS. And then, due to the differentiation of these two viruses, we understand that that was a new. And you can see here the world MERS outbreak. And you can see that the outbreaks still can be seen in the mentioned countries. And you can see that how the uh, number of cases rise within 2014, and then it still eventually go up. So more and more cases we see, and by the end of the 2020 we had documented 2,000 and a half cases over the in South Korea and Saudi Arabia and other countries. And up to now, we have around 807 deaths. So, and then the number of cases drop here, but then step by step, so the amount of infected people rise. So, MERS appear to be also zoonotic virus. So, can you see here a correlation? So, and we believe that MERS COVID-12, yes, is transmitted between animals and peoples, and the humans are infected through the direct or indirect contact with some animals and most probably camels. And then in humans, the virus has a strong tropism for non-ciliated bronchial epithelial cells. And this tropism is unique in that most respiratory viruses target ciliated cells. Uh, and it effectively evade the innate immune response. And maybe because it learns how to antagonize interferon production in the infected cells. So that's how we downregulate the development of the interferon in the body. So the MERS-CoV has dipeptyl peptidase 4, also known as CD26. That is a functional cellular receptor for MERS. So it has high affinity to this receptor. And that is the specific, and that is the difference between MERS and SARS. So to summarize it, by the end of the or by the 2019, we had already had two killers, coronaviruses. These are SARS-CoV, but then and MERS. But then the history of this disease a bit different. So SARS-CoV infected only 8,000 people, killed 774, about 10% of them. And then the virus had been contained in 7.5 months, and no diseases appeared uh, since 2003. My dears, SARS-CoV disappeared from human population, and it was really a miracle how fast it had been consumed. And the uh, doctor in China, epidemiologist, who was responsible for containing the virus, right, to stop the virus spreading, then had been uh, sent to the WHO organization to work because everyone was really satisfied with her work. But then the MERS hasn't disappeared, right? So MERS has never stopped, but it is much rare than SARS-CoV within the 2002 and 2003. So since arising in 2012, it infects around 2,600 people. And then still the infections is going on at very low level, but still MERS didn't disappear from human population, my dears. So two very related viruses and absolutely different epidemiological features of the disease. So, but then finally, a novel SARS-CoV-2, by now this is appeared in 2019, and by now it infects more than 264 million people, my dears. Of course, when this lecture had been developed a couple of days before, I mean, the upgrade of the lecture, now the numbers are changing. It keeps changing very quickly. So, and by that moment, the around 5.22 million people, infected people, had been killed by SARS-CoV. That is a really natural born killer. And it is not going to stop as we understand right now. So now we can see the common features in the originating of new coronaviruses uh, uh, that that are able to cause disease in humans, my dears. We are talking on only those that can cause disease in humans. And these are genetically diverse coronaviruses. By then, they had been passed through the natural hosts. And please pay attention that mainly when the new coronavirus, human coronavirus, appears, so the natural hosts had been bats or rats. That's it. So it means that the human viruses by now didn't appear from another natural host, just bats and rats. But then the natural virus from those host bats or rats supposed to adapt or undergo adaptation in some intermediate hosts. And it is discovered that for first human coronavirus that appeared in the mid 1960s, the two intermediate hosts are found, the cows for one of them, and llama, or how do we call it, for another one. So we also may have some not really very mild viruses appeared in 2004 and 2005. They didn't cause any epidemic. But then now we have that new coronaviruses. They originate all from the bats as the natural hosts, and the intermediate animals had been different. So, civet cat for SARS-CoV appeared in 2002 and 2003. Camels that had been intermediate hosts for MERS coronavirus appeared in 2012, and pangolins. These are the animals, quite rare everywhere. Uh, but then they had been speculated or suspected as an intermediate host for SARS-CoV-2 in 2019. And all of those viruses that we mentioned used to cause respiratory diseases in humans of different severity. So those were not severe, but then with time, I don't know what is the main factor. So, but then the new deadly coronavirus appeared from the same mode of adaptation. Bats, and then intermediate animals, and then ability to cause disease in humans. So, originally, this virus was called 2019 new coronavirus. But later on, the International Committee of the Taxonomy on February 11, 2020 officially named it as SARS-CoV-2 because of a genetic similarity to SARS-CoV, that was the first one. SARS-CoV-2 caused respiratory illness that is named by WHO as COVID-19 from Corona Virus Disease 2019. It is highly contagious and primarily transmitted among people through respiratory droplets. But then, even within respiratory droplets, there is a difference between small respiratory droplets and large respiratory droplets that we discuss later on. But also, it is spread through contact routes. And now we think it can be spread also through food ingestion. And WHO declared the COVID outbreak a public health emergency of international concern and later confirmed it as a pandemic on March 11, 2020, my dears. That's exactly when we went to the quarantine. First time on March. So, classification of the coronaviruses. So you can see that it is Nidoviralis, that is the order, and the family coronavirus, then subfamily coronavirus. And based on the serology and phylogeny, we may have alpha, beta, gamma, and delta coronaviruses. You can see here the names of those coronaviruses, but then all those that we are interested in belong to the beta coronaviruses. All human viruses are here. And the SARS, MERS, and SARS-CoV-2 has 75 to 80 genomic similarity. And the only alpha and beta may infect humans. And SARS and MERS has little genomic similarity, just 50%. That's not so much. Now let's take a look at the structure of the coronavirus. And as far as we mention usually the Baltimore classification, so this is type 4 according to the Baltimore classification. And it is enveloped virus with the super capsid enveloped the phospholipid bilayer with the several types of spikes. This is hemagglutinin esterase. This is a transmembrane protein. Then it is lined from inside with membrane protein and also have envelope protein. So, but the major one is the spike protein that is exposed to the environment. Then we see it in a red color. So insert it into the lipid bilayer. Inside we may have nucleocapsid protein and RNA. So here you can see the spikes protein that are glycosylated protein and they are trimeric, and this is a clue player in the binding in the human body, and it binds to AC2 receptor on the host cell with the proteolytic activity of the core receptor, and it is around 10 nanometers in size, and they are distributed along the phospholipid bilayer, and one virion particle has around 100 trimeric glycoproteins on it. Then you can see here membrane protein, and it is believed that the membrane protein is the interferon antagonist. So it means it decreases the activity of interferon. So, and in the virion, we have around 2000 copies of this protein. And then we have very small integral, transmembrane integral, so-called envelope protein, or E. This is a viroporin that is so-called, and it is responsible for host cell lysis because it mediates the production of the perforations in the cell membrane. And around the, or for a virion, we have around 20 copies of it. And then inside we have the nucleocapsid protein and protein that follow the structure of the RNA. And the RNA is positive sense single-stranded RNA. And the you see here that the symmetry of the nucleocapsid is helical, and the nucleocapsid protein is tightly fixed with the viral RNA. And because it is positive sense RNA, because according to the Baltimore classification, it doesn't keep its own RNA-dependent RNA polymerase with it. So, few words about the genome. Repeat as positive sense single-stranded RNA, and it acts as messenger RNA, and it is quite a large one with around 30 kilobase in length, and a bit unusual that it has 13 open reading frames, and quite a lot of non-structural proteins may be read from these open reading frames. Genome is considered to be longest in among the RNA viruses genome. So that's why maybe it encode so many proteins and non-structural proteins. So the single-stranded RNA of the coronavirus consists of 29.9 nucleotides, and it encodes just four structural proteins. These are S, you can see here, spike protein, small envelope protein, membrane protein, and N protein. And apart from this, around 15, 16 non-structural proteins are produced. Can you imagine? Just four structural and 15, 16 non-structural proteins. That make it, I think, partially so adaptogenic. So a few words more about the structure. Spike protein binds to the host cell receptor, and you should know what ACE2 stands for. This angiotensin converting enzyme 2 receptor. And S1 is responsible for vital function to regulate the virus host interaction. And S2 mediates the membrane fusion and viral entry. Then M protein involved in viral integration. Then the E protein is a virulence domain that activates immunopathological response. That's this in. And it facilitates assembly, envelope formation, and bonding. And the N protein has critical role in encapsulation of the genome, packaging intervariants, and effective in viral replication. That are the functions of those structural proteins. You can see here that the viral S protein, this is spike protein, can exploit other host factors to mediate viral entry. For example, vimentin is a cytoskeleton protein and lectin. So these are different target receptors that may somehow support the virus entry and multiplication. But of course, I would like to repeat that the best, very important target for this virus is ACE2 receptor. And this is the whole structure of this receptor. And this is the receptor binding domain. And it coincides with the particular region in the structure of the spike protein, my dears. This interaction is highly specific. Please remember that except for AC2, we may have two more targets in the cell receptors: vimentin, a cytoskeleton protein, and lectins. So a few words about the virus in replication. We have discussed the first step, the attachment of the virus, and then the second step is entry, and the third one is uncoating. You can see the process of binding, but then it is important that official host entry depends not only on the binding with SC2 receptor, but it also depends on the effective cleavage of the C1, C2 by the surface transmembrane serine 2 protease. And you can see this cleavage here. And it is also efficient when we have endolysosomal cathepsin L, which mediates the virus cell membrane fusion at the cellular surface and endosomal compartment respectively. So two proteins, transmembrane protease serine 2, right, on the surface of the on the surface of the cell, And when the virus is uptaken inside the cathepsin L, mediates the virus cell membrane fusion and release of the viral nucleocapsid into the cytosol that you can see here. So the one protein and another one. Then when the virus enters, the RNA genome is released into the cytosol, but RNA genome is still covered with nucleoprotein. It is not completely uncoated or naked. So the viral genome here is directly will be the stage of translation occurs, will be directly translated into the replicase proteins. These are the proteins that will help in virus replication. And the around two peptides will be produced, polypeptide 1A and polypeptide 1B, that are cleaved by the virus encoded protease. And then the process is called proteolysis, right? Once produced, those polypeptides are cleaved by the viral encoded protease. Then after the proteolysis, the polyproteins are cleaved into individual replicase complex non-structural proteins. These are these. So then we have further cleavage, replicase into the protein that performs the initiation of the genome replication. And these are catalytic subunit NSP12 and two accessory subunits NSP7 and 8 that increase the RNA-dependent RNA polymerase template binding and processivity, including the RNA polymerase. So they finally really it makes RNA-dependent RNA polymerase, but it consists of complex from several number of the proteins. Then what happens? How the replication happens. So the positive strand genome, this is still genomic RNA. It is used as a template for full-length negative sense RNA RNAs that act as subgenomic RNAs. And then some genomic RNA will serve for translation. My dears, do you see here that just a very small piece of genome is translated by now? So after the replication, when the subgenomic RNA is produced, negative strand, it will be responsible from the negative strand template. It will be responsible for translation of both structural proteins and non-structural proteins. So, that will be inserted into the EPR Golgi apparatus, intermediate compartment that is produced, and it is called RGIC. And the virion assembly will be performed here in this very special compartment after the translation of the viral protein. See what kind of proteins are produced. So see here, the four structural proteins are produced, N, S, M, E, and then the red one. These are accessory proteins will be produced, or non-structural proteins that are very important in the development of the virions. Then finally, the subsequent positive strand RNA genomes are produced and incorporated into the newly synthesized virions, or first the nucleocapsids. Then, when those nucleocapsids are produced and they enclosed with the RNA, then the process of viral assembly occurs when the virus, part of the Golgi apparatus membrane mainly. So, my dears, the origin of the supercapsid of or the envelope is not from the host cell but from the EPR Golgi intermediate complex membrane. See? So the membrane envelope and S proteins are incorporated into the EPR membranes, but not really cell membranes. And then the fully made or fully assembled virus particles will be secreted by endocytosis mainly. That is the way of virus replication. So, a few words about the disease because that is also of utmost importance. You know that most people infected with COVID-19 virus will experience, if it is symptomatic, because some cases may go asymptomatic, experience mild to moderate respiratory symptoms. Uh, and usually people recover without requiring any special appearance. See? So on the WHO poster, right? They are often mild, and but then it is spreading very quickly. Then older people, some children, and those patients with underlying medical problems may have very severe uh, lethal illness. And what kind of medical problems may make the disease more severe? Cardiovascular diseases, diabetes mellitus, chronic respiratory illness, and cancer. Those patients more probably may develop serious illness, and of course, immunocompromised individuals are all also at higher risk. So, uh, time course of the disease. So we have incubation period, and this is the period in between the contracting the virus and the appearance of the first symptoms. And usually we thought it is very long, but now we think it is around 5 to 6 days. And usually after these 5, 6 days after initial exposure, the patient will develop symptoms. So the problem of this disease is that the infection with SARS virus gives a diverse range of clinical manifestation. And you can see the range of this clinical manifestation from very mild infection to moderate cases. And then later on within the disease, please pay attention, not from the very beginning. So the severity will increase with time, and they can be severe illness or critical illness. Uh, in case the patients develop mild or moderate disease and recover like this, yes, that is the outcome of the disease. And it appears that in those patients, the immunity is able to develop control response. And this control the response is capable of controlling infection. And the response in the severe critical illnesses is a bit different from normal immune response. It is believed that in severe COVID illness, the excessive immune response is developed. And this, not the virus directly, but the excessive immune response uh, leads to the organ damage and even possible death. The viral load peak usually is seen within the first week of the symptomatic infection. It rises in dramatically in pre-symptomatic phase, but then in the symptomatic stage it will be very high. And usually it is often detectable by day 14. Then please pay attention that the viral load is not so high from the very beginning of the disease. That's why it is recommended to perform um, PCR test, diagnostic test, not very early within the disease or not just after the a suspicious contact with infected individual, because virus need to grow to multiply and then to reach its peak. Few words about the epidemiology of the disease. How then not really so we discussed the pathogenesis of the disease starting from the source of infection and route of transmission. So route of SARS-CoV-2 transmission is usually via infected respiratory droplets. What is the common situation where we may contract it? During stay in a crowded area, or most commonly when we sit around the table with our family members or friends, eating, speaking, and exchanging with the respiratory droplets. That is the most typical. So, especially when it happens not in the open area, but in poorly ventilated indoor settings. So contact direct or indirect. So this is a close contact route when the droplets. Then contact through fomite routes. So first the infection happens with the conjunctival nasal and other mucosa. Then fomite route that also happens through the conjunctival or nasal mucosa or through the GIT. So then the virus remains viable for many days on smooth surfaces. Do you remember at the very beginning of the epidemic, we were thinking whether we may contract this virus from the some parcels from Alibaba or whatever. So now we know that it stays viable for many days on smooth surfaces like plastic, stainless steel, glass, and at lower temperature and humidity. Then it feels very well. So, but then now we know that it is readily inactivated by commonly used disinfectants. That's why it is so important to treat all those parcels that we get and all those goods that we order and get through internet. So we have to treat them with disinfectants. And this is very, I would like to underline this high potential value of surface cleaning and hand washing. Because, see, this only partially infects directly from person to person. Then pay attention at the fomite route. So through hands, and then also we may get long range airborne routes with the respiratory droplets larger than what we have for the close contact. Pathogenesis of COVID-19. The roughly mortality rate of COVID-19 is estimated at 3%, but the mortality rate of critical patients was at one point as high as 61.5%. And usually lymphopenia can predict pneumonia development and progression to respiratory failure in patients. So uh, the cell counting in leukocyte formula is very important at that stage. Then, within the pathogenesis of COVID-19, hypercytokinemia-related immunopathology may serve a fundamental role of severe COVID. I hope you understand this topic, this word. So my students know that this is sort of Chinese language. Hyper too many cytokines, anemia in the blood. So this highly elevated number or amount of cytokines in blood, anemia, it is blood, right? So hypercytokinemia related immunopathology, that is the clue in understanding of the damage to the host tissues, because it is characterized with elevated plasma levels of tumor necrosis factor alpha, interleukins 2, 7, 10, granulocyte colony stimulation factor. Do you remember that is important in B cells activation? Then interferon gamma induced proteins 10, monocyte chemoattractant proteins, macrophage inflammatory protein 1 alpha, ENC reactive protein. My dears, those are, by the way, produced by produced by T helper cells, right? So now, the virion replication. The virus binds with the ACE receptor. This is a virus, right? So the virus binds with AC2 protein of the target cells, and virus replicates in the cells, and then it is released extracellularly. So there's a newly synthesized virus particles. Then the immune hyperactivity, inflammation signaling molecules are released by the infected cells and many induced organs. So the organs like liver, kidney, and spleen, then heart, and the initially, of course, lungs. These are infected, mainly infected organs. And the pulmonary destruction and multiple organ dysfunction, and this is multiple organ dysfunction, and these are organs where it is developed. Uh, is developed due to the disbalance of the activity of the cytokine C. So the cells of innate immunity will be activated. Dendritic cells, macrophages, and polymorphonucleated cells, right? They start expressing the damage-associated molecular patterns and pathogen-associated molecular patterns. And cytokines released from these cells are the oxygen-dependent radicals, right, and nitrates that may cause the damage of the cells. And then from the acquired immunity, when dendritic cells cause activation of T and differentiation of T and B cells and overproduction of antibodies, E and cytokines produced by T helper cells. Then it activates the T cytotoxic lymphocyte that also play a role in the tissue damage, and also they may mediate the production of T regulatory cells that can cause immunosuppression and secondary infections. And that also will be responsible for multi-organ dysfunction. The initial critical stage of COVID-19, when the virus enters the host cells, replicates, assembles, and release, and the replication directly causes the damage and destruction of parenchymal cells, such as alveolar and the epithelial cells initially, right? Alveoli. Then, at the same time, a large number of pathogens, associated molecular patterns, and pumps and dumps molecules released to stimulate the innate immunity response. It is stimulated and induce inflammatory cell infiltration, release large quantity of cytokines, chemokines, or proteases and free radicals, causing acute respiratory distress symptom, sepsis, and multiple organ dysfunction. Host cell receptors, these are angiotensin here, right, and in the gut, and as a result, the cytokine released will be interleukin 1 beta, 2, 6, 7, 8, 17A, gamma interferon, and granulocyte monocyte colony stimulation factors. They are all here. Then you can see the release of the macrophage inflammatory protein, tumor necrosis factor alpha, C-reactive protein, lactate dehydrogenase, then ferritin and dimer. These are the cytokines that cause so-called cytokine storm. So when the SARS-CoV viruses are sensed by the immune cells, such as macrophage, dendritic cells, and monocytes, their recognition results in production of pro-inflammatory cytokines that also damage respiratory epithelial cells of the lungs. Then, except for the acute respiratory distress symptom and extrapulmonary infestation, the COVID-2 may cause extrapulmonary diarrhea, shock, severe renal and liver dysfunction that we label as or mention as multiple organ dysfunction syndrome. So, by the way, if we are talking about pneumonia caused in the lungs, it includes bacterial acute changes, bilateral acute changes with diffuse alveolar damage, and vascular congestion, patchy inflammatory cellular infiltration, intraalveolar edema, hemorrhage, proteinaceous exudate, and denudation and reactive hyperplasia of pneumocytes, as well as the presence of multinucleated giant cells that may act as cytopathic effect of the virus. Then that is the simplified scheme of the so-called cytokine storm that, and the outcome is the RAD, multiple organ failure, hyperinflammation syndrome, and death. So when APCs are activated, that are macrophage and dendritic cells that lead to excessive release of the pro-inflammatory cytokines. We mentioned them already, right? And some chemokine that called cytokine storm. And then the damage occurs of the epithelial cells of the lining, and they reach into the bloodstream. So we have hypercytokinemia that we have discussed, right? So elevated level of pro-inflammatory pro-inflammatory cytokines in the blood. Then it may spread through the blood circulation, where it causes damage to other organs. So, what kind of immune responses are critical? Th1 mediated immune response plays a predominant role in the adaptive immunity against viral infection that you know already, Th1, right? So, and it depends mainly on the antigen presenting cell mediated cytokine environment. ENCD8 positive lymphocytes are capable of secreting gamma interferon granzymes, perforins, and also they are essential in eradication of the virus infected cells. B cells, uh, then the CD4 positive T helper cells facilitate the overall adaptive response by assisting cytotoxic cells. That's we know already. And the B cell mediated humoral immune response plays a protective role by producing the so called neutralizing antibodies. So we consider that mainly Th1 is immune response responsible for cytokine storm. Summary of the immunopathology, my dears. So you didn't study immunity yet. So that's why I try to describe the immunopathology in details, because that is the basis for tissue damage, not directly virus. So that's why you need to understand a bit of the uh, immunology right now, right? Okay. So, uh, regarding the summary of the immunopathology, depletion and exhaustion of lymphocytes. So the infection of the virus directly impaired the T cells. So then infected lymphocytes undergo depletion and exhaustion. We have as a result lymphopenia, anti-cell impaired, and dysfunction. And also the increase of the neutrophils. Uh, as a result, then activation and recruiting of the neutrophils also very important in the immunopathology. Then the over release of the interleukins that we mentioned already regarding T helper 17 that recruited monocytes, neutrophils, and macrophages that produce IL-1 and IL-6 also cause the cytokine storm. And the antibody, that is good that they are produced. But by the way, the binding of antibodies with the virus may cause antibody dependent enhancement or over the disease progression, because the virus not completely neutralized may cause the entry into the cell and the replication inside the cell, so and progression of the disease. So all this causes very poor outcome, the poor prognosis and outcome of the disease. Clinical features. So you know a lot, I think, about the clinical features, my dears. I would like to finish lecture because of the late start. I will go on with the lecture until I finish it. Then, if you will be not able to listen it right now, then please read it later on. Uh, we range the symptoms according to the uh, we rank them according to how often they are produced. So, on the first place, of course, fever ranks first because it is seen in more than 80% of cases. Then, cough is very close to it because it is seen in up to 60% of cases. Fatigue, 35% of cases. Sputum production is not very typical, my dears, just in around 30% of cases. And shortness of breath, more than 15% cases. The primary cause of mortality are respiratory failure, up to 70%. Then sepsis of multi-organ failure, close to 30%. Cardiac failure, 14.6, and renal failure, 3.7. These are most common cause of the. Then here you can see the computer tomography scan. And here you can see abnormalities that occur in 86%. The most common patterns observed in the scan. With ground glass opacity, this is it, and this ground glass opacity. Patchy bilateral shadowing. These are the shadows. Laboratory findings, as we discussed in the very beginning of the lecture. So the lymphocytopenia is seen in 83%. So around here, within here, thrombocytopenia 36% and leukopenia 33.7%. And please, we mentioned that we have very high level of C-reactive protein. Then alanine transaminases and aspartate aminotransferases are also high. Laboratory diagnosis. My dears, we have several levels and several possibilities of the laboratory diagnosis. So first, most commonly, the sample is the secretion taken from the oropharynx, nasopharynx. And here you can see the so-called express methods or rapid testing, because now it's so important to develop them specific and sensitive enough to perform it during the within the airports or before some meetings and so on, right? Usually it is quite often used in the airports. This is rapid testing. So if SARS coronavirus is there, so the within 10 minutes we would see the visual color change. Then if the nanoparticles are produced with the help of the reactives. So we may see naked eye detection. When we see the aggregated, these are gold nanoparticles. If we see it aggregated, then the reaction is positive and it can be seen by a naked eye. My dears, now we have a lot of this kind of testing. But then, and that is not laboratory diagnosis, because really it can be performed not in the laboratory, right? So this, I would say, this pre-lab diagnosis. Then how about the lab diagnosis? Usually the testing is performed by reverse transcriptase PCR test. So qualitative, Q stands for qualitative. Usually swabs are taken from the nasal, nasopharyngeal, oropharyngeal route, or sputum can be taken, or lower respiratory tract, aspirates or wash. Then positive test indicates the presence of the SARS-CoV-2 RNA, and together with the clinical picture, it supports the diagnosis SARS-CoV-19. Negative test result do not preclude SARS-CoV-2 infection and shall be interpreted in light of the clinical picture and epidemiological information. Serological tests. They are performed to detect IgM and or IgG antibodies against SARS in serum. The interpretation of the tests depends so much on the history of the uh, symptoms development or contact with someone also. The test can access prior exposure to the virus and cannot be used in the diagnosis of the current infection if we find the antibodies. So how it is performed? Uh, Usually, quite often, we use ELISA immune test or immune chromatography. You know ELISA beta, and that is the immune chromatography test. It is useful when the viral testing is not available. And also when patients with late disease complication need to make immediate decision in treatment, and the viral test takes more time. And in some patients uh, where the virus shedding is produced, the viral shedding dark the viral shedding is reduced, and making reverse transcriptase PCR result will be false negative. That's very important. So we can escape the the false negative result. Sorry, I had the telephone call. Now I'm free. So, rapid antigen testing. This is performed with the help of monoclonal antibody against SARS-CoV-2 nucleocapsid. Why nucleocapsid? Because usually nucleocapsid protein don't undergo that many antigenic variations. So that's why it is of utmost importance, because the tests directed to the surface spikes may be not highly sensitive or sensitive enough. So the N protein is abnormally expressed in infected cells usually. So monoclonal antibodies are specifically detected against nucleocapsid protein. So this is indirect assay, right? Because we perform. Do you remember? No, my dear, we didn't discuss. You didn't have immunology. This is capture sandwich assay. When we use known capture antibodies against the virus, then add the specimen taken from the patient with nucleocapsid protein. Then we add monoclonal antibodies to reveal the only N protein. And then we use the universal anti-human antibodies labeled with some substrate, some with the enzyme, and give substrate to reveal the visual reaction, right? So, and then it is possible to detect SARS-CoV-2. And it is believed that test has very high sensitivity, 86, but the specificity is even higher, 98%. So, how to treat the patients? 1 minute, I should answer the call. So we may distinguish several types of therapy: virus-based, immunotherapy, and uh, at least these two, right? So virus-based therapy. Uh, so you know that protocols for the treatment had been changed dramatically within this up to 1.5 years, right? So now what now they recommend? WHO recommend. Remdesivir as an inhibitor of viral RNA-dependent RNA polymerase, and lopinavir, that is the protease inhibitor developed not against COVID but against other infection, for example HIV, but it had been used to treat the COVID-19 infection. Uh, the non-steroidal anti-inflammatory drugs like cyclooxygenase inhibitors such as hydroxychloroquine or, or chloroquine phosphate are recommended to be used, and they are commonly employed in the management of SARS-CoV-2 infections, my dears. But we don't know exactly whether they are highly effective or not, because a lot of studies had been performed, and the results of those studies are quite controversial. So, but then you can see here with red letters that the use of this at least hydroxychloroquine in some studies associated with the finding of severe adverse effects like acute organ failure, opportunistic infections, and acute respiratory distress syndrome. Here you can see the common targets in the process of virus replication for virus-based therapy. So the polymerase inhibitors. So then the RNA dependent, the inactivation of RNA dependent RNA polymerase interfere with the minus sense RNA production that's supposed to be used as a template for viral genome replication. Then protease inhibitors interfere with the cleavage of the first and second types of protein that are produced before and after the genome replication. Then uh, hydroxychloroquine. This, we believe, it interferes with the uh, fusion from endosome, fusion of the virus with the endosome. We think it interferes with the acidification of the endosome. And also now a lot of research groups that are working on the entry inhibitors. But we don't see any very well studied and licensed drugs for it yet. So, but we can see that there are some structural targets in the virus itself and inability to prevent viral entry by blocking the ACE inhibitors. the very important treatment is convalescent patient's plasma. That is a very good thing. So now we take a look at the immunotherapy of COVID. So we may have interferon-based immunotherapy, where the interferons alpha, beta, and lambda can be used in treatment. Then, antibody-based therapy, where we may use plasma. Then, if it is highly purified, we may use intravenous immunoglobulins, or we may use monoclonal antibodies. These are approaches that now are in the process of development. First two are developed already. Then in immunotherapy, we have the approach that is cytokine storm management. So what does it mean? It means we may act in two ways. First way, we may block cytokines. Cytokine blockage. These types of cytokines supposed to be blocked. IL-6, 1, 17, tumor necrosis factor, interferon gamma, and we may perform also complement blockage. Then the new approach is cell therapy. So when we're supposed to use the cells trained against coronavirus to cause uh, the replacement of the damaged cells with this. Then we may have immune check, but that is in process. Immune checkpoint blockage of the specific types of cells development regarding using the particular types of cytokines. Then we may have active immunotherapy. What do we mean? This is SARS-CoV-2 vaccine, and there are several types. I would of course, they are used for immunoprophylaxis, but then maybe once it can be used for immunotherapy. And then it is believed that some injections or some previous infections may improve the immunity, so it will fight better with the coronavirus. As one of the possible targets is BCG vaccination. I think you might heard about it, that previous BCG vaccination performed within the lifetime will improve the immunity against COVID. It is not proved yet, my dear. That is then something. Then we have other quite sophisticated types of immunotherapies. You take a look at it later on. Now, so that what we have right now. This is the interferon-based therapy. So these are the recombinant interferons that are recommended. And here we have type 3 group of antiviral cytokine that consist of 4 interferons lambda molecules called interferon lambda 1, 2, and 3. And they are also known as interleukins. And interferon lambda 4. Their functions are similar to that of type 1 interferon that have direct antiviral activity, but they are less intensive and serves mostly as a first-line defense against viruses in the epithelium. So they're supposed to be used in early stages of the infection. Then for antibodies, of course, the primary one is a hyperimmune convalescent summer source. So, and it restricts viral infection and modulates the cytokine storm, which finally improves their respiratory function. and it improves also outcome in COVID patients. Monoclonal antibodies. So this is the creation of human monoclonal antibodies that can neutralize SARS-CoV-2 from B cells of convalescent patients. So, my dears, this is produced in human cells, not in mouse cells. So, usually these antibodies target the S1 subunit responsible for attachment to the attachment of critical receptor. And then intravenous immunoglobulins, polyvalent usually. And they work through several mechanisms to fight with anti-inflammatory effect. The inhibition of C gamma receptors, neutralizing cytokines and antibodies, suppressing the activation of lymphocytes, and inhibit the function of complement system. That is the activity of the polyclonal antibodies taken from the blood of reconvalescent patients. Then we may manage the cytokine storm. So these are the critical, this is the critical cytokine, interleukin 1 and interleukin 6. They're supposed to be blocked with the specific antibodies against these interleukins. And these antibodies inhibit the inflammatory pathway induced by interleukin 6, and this is tocilizumab and sarilumab and anakinra, and is a recombinant interleukin 1 receptor antagonist that block the interleukin 1 receptor in the body. In severe stages of the disease, they could be an efficient approach to reduce the progression of the cytokine release syndrome and ear distress syndrome. Respiratory distress symptom, acute respiratory distress symptom. Then interferon gamma blockage, that's also important, and this is emapalumab that inhibits the progression of the RDS. Immunosuppressive agents, corticosteroids, out of all, dexamethasone is recommended because it reduces the concentration of interleukin 8 and 10 after administration. So it inhibits the expression of the pro-inflammatory transcriptional factors in the nuclei of inflammatory cells. And it is not supposed to be used outside the hospitals in mild and moderate diseases. In very severe, only because it causes immunosuppression. So vaccination against coronavirus. Here you can see the all really very common vaccines and approaches used for vaccination. So the live attenuated vaccine is not really very common approach, right? Because we believe it may, under some conditions, cause the cytokine storm, so this is not very popular target. But the virus itself may be used in inactivated virus-based vaccine. So, for example, COVID-19 that can be performed in Russian Federation, COVID-19, right? Then the virus is killed and all proteins of the virus can be used in immunization. And it is good because the all viral proteins will be uptaken by dendritic cells and presented and antibodies against all antiviral proteins will be produced. The very popular are nucleic acid vaccines. When the RNA directly or DNA can be used for injection into the human body, and they will be uptaken and the peptides will be produced, and then the vaccination will occur. Then the viral vector vaccine. This is a based for Sputnik, for example, adenovirus vector uh, incorporated with the specific gene that code for the spike glycoprotein. So it also can be used. Then protein-based vaccines, when the subunits of the virus are already isolated, purified, and used directly as the antigen. That's a most uh, traditional approach, right? And then virus-like particles that are developed out of the carrier and the peptides that mimics the spike protein of the virus, and usually they are used together with the adjuvant, for example, alum, because they are poorly immunogenic. And in Russian Federation, Epivac corona peptide vaccine is performed regarding this approach. So, Covivac inactivated by beta propiolactone and also administered with aluminum. Then Sputnik V is adenovirus vector vaccine of the serotype 26 adenovirus. And serotype 5 for the first component and serotype 5 for the second component. So why two different distinct serotypes are used? Because when this viral vector is introduced into the human body, the immunity will be developed not against this spike protein of the coronavirus only, but against the adenoviral vector. So the adenoviral vector immunity, even if it is very low, it can be developed. Then, to overcome the effect when of the secondary immune response when the same serotype is produced. So the second component is based on the different serotype not neutralized by serotype 26 antibodies. So, Epiva corona we described already. What are the disadvantages of Sputnik vaccine? Anti-vector immunity, existing anti-vector immunity, because these serotypes are circulating in the human population. So that's why we use two types of the vectors. So, some more COVID-19 vaccines. For example, Moderna uh, and the National Institute of Health have jointly developed mRNA-based vaccine consisting of the six sequence optimized messenger RNA encoding the spike glycoprotein encapsulated into the lipid nanoparticles to improve the entry of these nanoparticles or uptake of these nanoparticles by dendritic cells and macrophages. So we need dose in dose escalation trial. This vaccine induced both spike glycoprotein binding and virus neutralizing antibody response in a recipient aged from 18 to 55 years. Then the second one, Pfizer and BioNTech have developed a series of mRNA-based A-lipid A nanoparticles formulated again. The nanoparticles are used for better uptake of the RNA by the antigen presenting cells and other cells. This nucleoside modified mRNA vaccine elicit uh, uh, red blood cells binding of IgG and neutralizing antibodies with mostly mild side effects. What side effects? Injection of inside of injection site pain, fatigue, headache, chills, muscle pain, and joint pain. So Usually it is given twice. So that is the type of Pfizer-BioNTech vaccine made up of mRNA. So, and it should be encapsulated in a lipid coat. Otherwise, it will not be able to enter the human cell. The disadvantage is that this vaccine is supposed to be stored at -60, -80 before use. Okay, my dears, with this we finish, and thank you very much for your attention. So I hope it will be enough to know more about the coronavirus itself and coronavirus infection. This exam is also placed into the exam card, so please, we also will have a class for SARS-CoV-2 infection. See you in the classes, and next lecture will be after 2 weeks. We have lecture once in 2 weeks. Goodbye, my dears. That's it.